You are standing in the middle of King's Cross with a postcode in your hands, your feet trodden on by the busy crowd. So much has changed since you were last here 20 years ago, and everywhere you go seems to meet frowning faces set in their own path. By a lucky coincidence, somebody recognizes you and shakes your hand. They are going the same way. What are the odds? Thank you, you mutter. You are a star. Through thistles and thorns, spinal cord injury leaves patients isolated from their own bodies with devastating lifelong consequences. The limited nature of adult human spinal cord repair is frustrating but understandable from a developmental point of view. If fasciculus gratulus, a tract of the spinal cord responsible for lower limb sensations that relays touch from the foot, gets traumatically disrupted, the dorsal root ganglion DRG cell axon would not only have to cross over the site of injury, but also find the correct path all the way up to 50 centimeters in a tall individual. Even if that tremendous task is accomplished, there still remains the challenge of finding the correct second-order neuron of the nucleus gratulus that, in turn, connects to the third-order thalamic neuron transmitting signals to the part of the somatosensory cortex representing the foot. Not something that can be easily done without help. On the way to connectivity restoration, it has been noted that the three main aims of axon regeneration research are to initiate and maintain axonal growth and elongation, to direct regenerating axons to reconnect with their target neurons, and to reconstitute original circuitry or the equivalent, which will ultimately lead to functional restoration. Although many interventions have now been reported to promote functional recovery after experimental psi, none has fulfilled all three of these aims. In fact, only the first aim has been extensively studied and convincingly demonstrated. Indeed, even though the possibility of axon regrowth in the adult mammalian nervous system has been shown, the evidence supporting neuronal connectivity restoration is rarely convincing. Without careful guidance, aberrant axonal regrowth is a serious obstacle to functional regeneration. Interestingly, long-distance regeneration of axons is not the only mechanism through which normal spinal cord function can be restored. Injury is known to induce plasticity by several mechanisms, including unmasking inactive spared pathways, compensatory collateral sprouting from the intact pathways, or an increase in receptor number due to denervation hypersensitivity. For example, Hofstetter al. commented that excessive local sprouting in our present study might have facilitated the formation of novel corticospinal tract pathways, although we could not detect a correlation between the amount of local sprouting and motor recovery. Whilst researchers are trying to find a way to artificially guide axons to their correct targets and induce plasticity, there is a cell type that routinely orchestrates these processes. Astrocytes express a range of axon-attractive and repulsive molecules that are crucial for proper development and adult nervous system plastic reorganization. They provide a physical adherent substrate for growing neurons, secrete extracellular neuroattractants like vimentin and repellents like chondroitin sulfate proteoglycans CSPGs and semaphorins. A scar or a star. The neurocentric paradigm considered astrocytes to be a barrier to healing after the psi for almost a century, despite the lack of evidence that purely neuron-based therapies are sufficient for full regeneration. However, if the derogatory dubbed astrocytic scar is removed or prevented from forming in the psi context, not only do axons fail to spontaneously regrow through the lesion, they also become unable to regrow upon delivery of stimulating growth factors that promote growth in the presence. The usefulness of intraspinal neural stem cell grafts directed differentiation improves outcome. Nat neurocyte 200,583,346-53. DOI 10.1038NN1405. Allen N.J., Bars B.A. Signaling between glia and neurons. Focus on synaptic plasticity. Volume 15. Current Opinion in Neurobiology, 2005. p. 542-8. DOI 10.1016J.CONB.2005.08.006. Freeman, Mr. Sculpting the Nervous System. Glial Control of Neuronal Development. Kerr Open Neurobiol, 2,006,161,119-25. DOI 10.1016J.CONB.2005.12.004. Fallon, Jr. Preferential outgrowth of central nervous system neurites on astrocytes and Schwann cells as compared with non-glial cells in vitro. J. Cell Biology, 19851001198207. PMID 3,880,751. Shigeo M. Toda C. Extracellular vermentin is a novel axonal growth facilitator for functional recovery in spinal cord injured mice. Scirep. 20166 February 28293. DOI 10.1038 SREP 28293. Wang H., Category Y, McCann T., Unsworth E., Goldsmith P., UZX, al. Chondroitin 4-sulfation negatively regulates axonal guidance and growth. J-cell Psi 2,8,121,183,083-91. DOI 10.1242 JCS.032649. Molofsky A.V., Kelly K.W., Psi H.H., Redmond Sa, Chang Ed M., Madaradi L. al. Astrocyte-encoded positional cues maintain sensory-motor circuit integrity. Nature, 20,145,097,499,189-94. DOI 10.1038 Nature 13161. Chu T., Joe H., Li F., Wang T., Lu L., Feng S. Astrocyte transplantation for spinal cord injury current status and perspective. Volume 107, Brain Research Bulletin, 2014. p. 18-30. DOI 10.1016J.Brainsball.2014.05.003. Anderson M.A., Berdager, Ren Y. Al Y., O'Shea trademark, Kawaguchi R. al. Astrocyte scar formation aids central nervous system axon regeneration. Nature, 2,016,011-20. DOI 10.1038 Nature 17623. Tyzak G.E., Sitnikov S. Barson D., Adamskar K.L., Lau and K., Kwok J.C., al. Astrocyte response to motor neuron injury promotes structural synaptic plasticity via ST83 regulated TSP1 expression. Nat Commune, 201,454,294. DOI 10.1038 NCOMMS 5294. Marcius Mai, Searing MB, Pete CMA, Crow MJ, Alexanian R, Kirpad SN. Pain with no gain allodynia following neural stem cell transplantation in spinal cord injury. EXP neural, 20,062,012,335-48. DOI 10.1016 J, X neural .2006.04.035. Davies Zhe, Huang C, Proschel C, Noble M, Mayer Puschel M, Davies SJA. Astrocytes derived from glial-restricted precursors promote spinal cord repair. J. Biol, 2,006,537. DOI 10.1186 J. Biol 35. Davies Zhe, Proschel C, Zhang N, Noble M, Mayer Puschel M, Davies SJA. Transplanted astrocytes derived from BMP or CNTF-treated glial-restricted precursors have opposite effects on recovery and allodynia after spinal cord injury. J. Biol, 20,087,724. DOI 10.1186 J. Biol 85. Teshigawara K, Kuboyama T, Shigeo M, Nagata A, Sugimoto K, Matsuya Y, al. A novel compound, denosumab, ameliorates spinal cord injury via axonal growth associated with astrocyte-secreted vermentin. BRJ Pharmacol, 20,131,684,903-19. DOI 10.1111 J.1476538.1.2012.2.02211, X. Image via Stocksnap Ixabay. Further reading.